So today we'll be exploring some of the different molecular biology techniques that are used to study genetics. And I really like studying this chapter at the end of the semester. It ties together a lot of the things that we've learned so far, and we can better understand the goals of taking these techniques when we know what some of the different goals are in actually learning about different types of genes. Also, some of these molecular techniques can be combined with other techniques that we've studied so far, such as linkage analysis and pedigrees. So we would say that we're now in the middle of a molecular genetics revolution. Okay, this means that the speed of technology has been advancing very fast, and we've been using new techniques not only to advance our scientific knowledge, but also to change things that affect the general public. We've been able to use recombinant DNA technology, okay, otherwise known as genetic engineering, to join together DNA segments from different species or from different parts of the genome in the same organism. This has had major implications for developing new products and new organisms that have implications in both medicine and agriculture, as well as in other fields. Okay? So in this chapter, we're going to be talking about working at the molecular level. Okay, instead of just breeding organisms and looking at phenotypes, we're going to be actually looking at segments of DNA. Okay, so first, in order to study DNA, it's a lot easier to hone in on the gene of interest or to sequence DNA if you've cut your DNA into smaller pieces. Okay, it's a little bit hard to work with an entire chromosome or an entire genome. Okay, so we can use a variety of different types of products. Restriction enzymes, which are naturally occurring in bacteria. Engineered nucleases, okay, which cut DNA and are designed by scientists, so they have some flexibility over the sequences involved and where these enzymes cut. We'll also be talking about an exciting and relatively new process called CRISPR. The CRISPR-Cas genome editing system has been extremely effective and is showing a lot of promise in gene therapy or changing the genes of people that have genetic diseases so that they will basically become healthy. We will also talk about separating and viewing different DNA fragments. And then when we want to work with a particular piece of DNA, and we've got to figure out which one it is, we'll be able to either locate the DNA fragments with various types of probes, which are often complementary sequences, or we can use special techniques to identify bacterial colonies that have taken up a gene of interest versus not taken up that gene. Okay, restriction enzymes are enzymes that have been in use for a pretty long time in genetics. They're able to recognize and cut DNA at a specific nucleotide sequence. Okay, so usually these sequences are around 4 to 6 base pairs long, and they're going to be cutting anywhere they see that sequence. Okay, so if we're looking at a circular piece of DNA in a bacterium with a small genome, one restriction enzyme may cut several times. In a larger eukaryotic genome, they may cut thousands of times. Okay, so that's one issue with using them. Okay, once these fragments are cut, they can be somewhat unstable. Okay, think about the ends of a chromosome. Linear DNA is less stable than the circular DNA that's found in the plasmids in bacteria. So we also have to add some methyl groups to the recognition sequence where the cut has been made, which is the new end of the piece of DNA. And that helps to stabilize the piece and prevent it from being digested in the enzymes that would normally break up DNA fragments in bacteria. When we make a cut with the restriction enzyme, okay, usually there are short single-stranded overhanging ends, okay? The restriction enzyme makes a jagged cut, which allows the cut piece of DNA to more easily recombine with other pieces of DNA. Okay, sometimes these cohesive ends are also known as sticky ends because of their ability to recombine fairly easily. We can also have restriction enzymes that leave blunt ends. Okay, that means that a single cut is made at the same point in both single strands. So this table here is not anything that you need to memorize, but I'm showing it because it shows examples of some of the commonly used restriction enzymes, and you can see some of the features here. Okay, we have 5 displayed here that show the cohesive or sticky ends, and they're going to be making the cut at a different place on one strand of DNA from the other strand. Okay, so they're going to leave some overhanging single-stranded DNA. 2 of these leave blunt ends. Okay, and you can also see the size of the recognition sequence. The recognition sequences are usually between 4 base pairs long and 6 base pairs long. So one of the issues with that is that in a large genome, you're going to have many fragments made, okay? Even just by chance alone, a sequence of 4 nucleotide bases is going to occur very frequently. A 6 base pair sequence somewhat less frequently, but still, in a large genome, such as the human genome, or really any eukaryotic genome, you're going to get a lot of cuts and a lot of pieces of DNA. But in a bacterium with a small genome, 2 to 5 cuts or something might be a lot more typical and make using these restriction enzymes a bit more manageable. Okay, this diagram shows what the pieces are going to look like after the cuts are made. You can see this enzyme here, HIND3, is making one cut here and another there, leaving an overhanging piece known as the sticky ends. Okay, this is actually very useful for biologists that want to insert another piece of DNA. Okay, if they use a piece of DNA with complementary ends, it's easily inserted and incorporated into this piece of DNA. And we'll talk more about why that's important in a moment. Other restriction enzymes that make a blunt cut leave these blunt ends without any overhanging pieces. Although this might seem a little bit counterintuitive, the blunt-ended DNA is actually less stable than the sticky-ended DNA. It's also a lot harder to join pieces of DNA to these blunt ends. So if the purpose is to make recombinant DNA, or to join a gene into this piece of DNA that comes from another organism, typically the enzymes that produce the sticky ends are preferred. This diagram shows how 2 pieces of DNA cut at the same location with the same recognition sequence can then be combined. The blue piece and the red piece come from different sources. DNA ligase, okay, do you remember that enzyme from an earlier chapter? Is very good at joining the single-stranded breaks, okay? So it's very easy for them to join these sticky ends together. This enzyme can sometimes be used to join blunt ends, but at a much lower success rate. Okay, let's just review for a moment. Where do restriction enzymes come from? Well, they've been isolated from bacteria where they are naturally occurring. Okay, and these bacteria typically use them to cut up foreign pieces of DNA that are introduced by viruses. Okay, we can also use engineered nucleases. These are enzymes that have been created by humans to make double-stranded cuts in DNA at specific sequences. Okay, one of the reasons these are important is because sometimes we want to cut DNA at fewer sites than restriction enzymes would cut them. So think for a moment, would we want a longer or a shorter recognition sequence if we want to make fewer cuts, okay, and be dealing with fewer larger pieces of DNA? Okay, hopefully you said longer sequences. Okay, it's less likely that 20 particular base pairs will be arranged in a certain way at many points in a genome than a sequence of only 6 base pairs. Another really useful technique for creating recombinant DNA is the CRISPR-Cas genome editing system. This was discovered in 2012 as a naturally occurring system in bacteria and archaea. So there's a protein and a piece of RNA that locates a sequence complementary to that piece of RNA and breaks the DNA there. Some of these naturally occurring CRISPR proteins have been able to be modified for scientists to use in genetic editing. They seem to be a lot more effective and efficient than the previously used systems to try and edit DNA in patients who have suffered from a particular genetic disease and desire to change their genotype. So the CRISPR protein recognizes a sequence complementary to the RNA and cuts it at a specific sequence. The cell then has mechanisms for repairing the double-stranded breaks, therefore editing the target sequence. Sometimes we just want to destroy a defective gene. Okay, by the time the repair is made, usually those ends are unstable enough, they've been degraded, and they can't make the target protein that's causing the problem. Other times, a new piece of DNA is introduced, and the defective gene can be replaced by the new one. So this diagram shows the Cas9 protein in yellow here, and a particular piece of guide RNA that helps it to locate the place on the DNA where the repair needs to be made. Scientists can use whatever RNA sequence that they want that's complementary with the DNA sequence that they want to target. Notice how this complex of protein plus RNA makes a break, and if the scientist inserts a new piece of donor DNA at the particular time, it's likely that that piece of DNA will be incorporated into the genome. To this date, this has been the most effective system that we've discovered for inserting new pieces of DNA and getting them to apply to a large number of cells. When we're working with bacteria, it's okay if we want to try to get the bacteria to uptake a particular gene and it only works a small percentage of the time. We only need to find one particular cell in which it worked and then allow that cell to grow over and over again, since bacteria can replicate very rapidly. In contrast, if we're trying to help a patient that suffers from a genetic disease, we need a much more efficient system that's going to work more of the time. Okay, since we want to repair that gene in most or all of the patient's cells. Although using this in a clinical setting is still in the experimental trial system, it appears that it's a very promising technique.